Dear teachers and esteemed audience, good day to all of you. My name is Zhang Qi, and today I will be providing an interpretation of a recently published article in iMeta. Akkermansia muciniphila administration ameliorates streptozotocin-induced hyperglycemia and muscle atrophy by promoting IGF-2 secretion from mouse intestine. Type 1 diabetes mellitus, T1DM, characterized by the destruction of pancreatic beta cells. Large-scale epidemiological studies have explored the changes and potential roles of gut microbiota in the pathogenesis of T1DM. Akkermansia muciniphila is a prevalent mucin-degrading gram-negative bacterium. Recent studies have highlighted the positive effects of A. muciniphila on host metabolism and immunity. In our present study, we employed A. muciniphila gavage to investigate the physiological roles and molecular mechanisms of A. muciniphila in the progression of T1DM. We subjected mice to a single dose of 100 mg per kilogram STZ injection and kept them until 5 mpi, month post-injection. The fasting glucose levels of the mice kept increasing after STZ injection and peaked at 4 mpi, while significantly decreased at 5 mpi compared to those of 4 mpi. PCA analysis of 16th rRNA sequencing from cecal contents revealed a clear separation of STZ-injected groups with control group. The Shannon index indicated bacteria community diversities were significantly decreased at 2.5 and 4 mpi, and partially recovered at 5 mpi. Rate Bacteroidetes/firmicutes was also significantly decreased at 4 mpi. At 5 mpi, bacterial communities involved in insulin signaling pathway was found upregulated. Metagenomic next-generation sequencing showed that A. muciniphila was in the top fold-changed bacteria species between 4 mpi and 5 mpi. The mRNA levels AX1 was significantly upregulated at 5 mpi from intestine. Wild-type C57BL/6J mice were then orally administered for a month before STZ injection with saline. Heat-killed A. muciniphila, AKK, sodium acetate, ACE, and sodium propionate, PRO, respectively, Figure 2a. At 3 and 5 week post-STZ injection, body weights of mice from saline, ACE, and PRO groups were significantly reduced, while there was no difference in those of AKK group. The fasting glucose levels of the mice from AKK and ACE groups were significantly reduced compared to saline group, Figure 2c. In glucose tolerance test, GTT, mice from AKK group showed reduced glucose levels and improved glucose sensitivity. Metabolic analysis showed that A. muciniphila gavage significantly elevates the O2 consumption, CO2 production, and heat production of the mice. A. muciniphila gavage also reduced the water intake of STZ-induced T1D mice. Weight of different muscle tissues, including tibialis anterior, TA, and quadriceps, CHU, were all larger from mice of AKK group compared to those of saline group, Figure 2j, K. Cross-section of TA also revealed that average fiber size, Figure 2L. Protein level of MSTN all significantly reduced from TA of AKK mice. The expression levels of CPT2, PARC1 alpha, COX7A1, AX2, HK2, LDHB, SLC2A1, IRS2, INSIG1, FNDC5 and MYH2 were found upregulated genes after A. muciniphila gavage. In addition, protein levels of ATGL and CPT2 were significantly increased in TA muscle from AKK group. To identify the key secret proteins that were responsible for A. muciniphila treatment-induced muscle mass, we extracted the DEGs under a same threshold from saline versus control and AKK versus saline, respectively. Pathway enrichment indicated that the 242 overlapped DEGs were mostly involved in imstim process, endosome. Particularly, IGF-2 was downregulated from intestine after STZ injection and recovered in AKK group. We verified both mRNA and protein levels of IGF-2 from intestine, and serum IGF-2 levels were increased. Instead, mRNA levels of I2R and IGF-2BP1 were significantly increased from in TA muscle of AKK group. Our results suggest that the administration of heat-killed A. muciniphila could be a potential therapeutic strategy for T1DM and its associated hyperglycemia.